One. Good afternoon. This is John Bennett, Dr. John Bennett in sunny Miami. We have another show for MedGadget.com. We're graced with the presence of Yakov Namayas. He's the director of the Grass Center for Bioengineering at Hebrew University, and currently he's in Boston working with MIT and Harvard, I understand, on sabbatical, and he'll explain that. And we're also joined with Gene Ostrovsky. First, we'll introduce Gene. Hello, Gene.
Hi, Dr. Bennett. I'm Gene Ostrovsky. I'm the managing editor of MedGadget. And Dr. Namias, thank you for joining us here. We're very excited to have you.
Okay, Kobe, it's all yours.
John, Gene, it's a pleasure to be with you today on MedGadget website, and I guess we're going to talk about liver on a chip today.
Yes.
Is that correct?
Yes. Yes.
Okay.
Please give us a summary of the technology you're working on, and where you see it going, and give us just an overview, and then we'll be asking you questions, regarding that, and see where it goes.
Sounds good. So you asked me to prepare a little presentation. So let me start with my presentation. So we've been working for a while on technologies to replace animal experiments in the lab, and our primary focus is the human liver. Now, the liver is my favorite organ. It's the largest internal organ in the human body, about 1.5 kilograms in an adult. It's essentially serves as a gatekeeper of the blood. Everything we ingest and is absorbed by our intestine finds its way to the liver, and the liver is tasked with homeostasis of different metabolites. So it will take carbohydrates and remove them quickly from the circulation after breakfast, for example. It will take the same carbohydrates like glucose and synthesize them into fatty acids and lipids, repackage those lipids, send them to the rest of the body, and then it will also use different metabolic enzymes to synthesize steroids and bile acids, playing a role as secondary messengers and help us ingest our foods.
Mm-hmm.
Now, because it has these different enzymes, yeah.
No, please go ahead.
Because it has these different enzymes, it's also the site of drug metabolism. So this is something that the liver hasn't really been evolved to do. We didn't have acetaminophen for millions of years. We had acetaminophen and different drugs just in the last couple of decades, but it's the same enzymes that break down secondary messengers and polyphenols that break down drugs. And when you look at drug development, especially today in the age of combinatorial chemistry, we find that the case is that for every disease we're trying to develop a drug for, we can easily find dozens to hundreds of candidate molecules. And the process of drug development today is essentially screening among these candidate molecules which one is effective at the smallest dose, and which one is toxic at the highest dose. And that's really our safety margin. And this is where all game in pharmaceutical drug discovery is being played. Now, if you do everything right, it takes you about a decade, and it costs you about $1 billion as cost starts to escalate from preclinical to clinical development and then to marketing to get the drug to the market. If you do something wrong and you don't estimate the proper toxicity, then failure in phase 3 trials or failure after you reach the market and you have to withdraw the drug can cost many billions of dollars to the company. This is why the pharmaceutical sector is really interested in trying to predict what is the toxicity profile of many chemicals and get a better handle on that. The biggest problem we have is that right now we can primarily use only animal models, especially for long-term toxicity studies that are needed for clinical trials. And animals like rats and mice are horrible predictors of toxicity. About 70% of the compounds that affect humans will not cause toxicity in mice and rats, and 70% of the compounds that affect mice and rats don't affect humans. Excellent examples are aspirin, which causes developmental toxicity in rodents, is very, very safe in humans, and analgesic drugs, for example, in the 60s, like Talamid, that were completely safe in rodents, cause severe birth defects in humans. This is why about several decades ago, Mike Schuller and others came with the idea of trying to mimic human metabolism on a chip, essentially creating artificial organs and growing them on chambers in microfabricated devices, and then using small microchannels to connect those chambers and to mimic human physiology. The idea is that you have human genetics in human cells, and you'll be able to do a lot of experiments at once, either with different types of cells or with different types of drugs, and essentially mimic human physiology on a chip. Now, it might sound to some like a little bit of a science fiction, but the first company to take Mike Schuller's technology and market it is called Urel, and it's been in the market for a while now. And I've been fortunate in my days at Harvard Medical School to help them develop the chip and the culture design for the liver cells. And you can see the chip in the right-hand corner where they have the liver in one chamber and cancer in another chamber, and essentially looking at rates of breakdown of drugs and how the metabolites affect tumors and other organs. Now, it's not a science fiction, and the question is, where have we been going in the last couple of decades? And one of the things that happened quite recently is the development of microfluidics and rapid proliferation of nanotechnology. Well, with microfluidics, we can do quite a lot of things. One of them is create a stable stimulation, essentially expose cells to a very precise and very stable concentration of any drug that we want or any chemical that we want. Now, it sounds trivial, but it's not. If you take a look here on the left side of the slide, you can see what happens when we do an experiment in a standard Petri dish. What happens is that the second that we add our chemical or hormones or growth factor, then the first thing that happens is that we have non-specific absorption causing anything from 20 to 30% of the chemical to disappear. And then rapidly after that, cells and the tissues start taking it up, or it starts binding to the cells, and you have even faster decreasing concentration of whatever chemical you're looking at. 24 hours later, a student is changing the culture medium, and you get to the same effect again. Essentially, you're thinking that you're measuring the cellular response to 1 nanomolar of a drug, but in reality, you're giving the cells a rapidly decaying signal. Well, if you perfuse it using microfluidics in a bioreactor, what happens is that in the first couple of minutes, all sites have become saturated, and you no longer have non-specific absorption, and you reach a steady state with the clearance rate. Essentially, that means that if you continuously perfuse a chemical on cells in a microfluidic system, then the cells will see the right concentration, the same concentration throughout the experiment. So this is very, very important for getting the right number. The second thing is that we can essentially mimic human physiology. We'll show you in a second. And then because we are doing things on a micro scale, we can do much less many less cells per experiment than a Petri dish, for example. Are you guys still with me, or did I lose you completely?
We're here, just on mute.
Yes, we're here.
So this is the bioreactor we designed at the Hebrew University and was produced in partnership with the Fraunhofer Institute in Germany. It's a microwell bioreactor, and you can see the wells here on the right. And it has a small glass slide that allows us to create organs in each one of those wells in an open configuration. And after 24 hours that we form these microorgans, we can seal them in the device and start perfusing them. Now, when you talk about the liver, it's important to note that the liver physiology, microphysiology, plays a critical role in its function. First of all, this is intervital microscopy of the liver, and you can see that it's very highly vascularized. Every liver cell called a hepatocyte has at least 2 capillaries encircling it. And there is a gradient of nutrients like oxygen and hormones that develop along the capillary from the entrance to the exit of the liver. And this causes the cells in the beginning of the liver, the periportal region, and the exit of the liver, we call it the perivenous region, to behave very, very differently. For example, the cells at the bottom, the cells at the exit, actually have much higher levels of drug metabolism enzymes called CYP450s, while cells at the entrance of the liver, for example, do a lot of beta oxidation, so they break down fatty acids. This is very, very important for liver function. The way we're mimicking it in our bioreactor is by allowing the cells to consume the nutrients into the well. So if you take a look at our finite element modeling, you'll see that there is a steady gradient of nutrients like oxygen, glucose, that happens into the well, essentially mimicking what happens in vivo. And then we can create very complex organs inside each one of those wells. And you can see on the left an image of our 9 wells and human microlivers that we develop. Each one of them is about 1 millimeter in size. And they can be composed from primary human hepatocytes and endothelial cells mixtures, or human embryonic stem cell derived stem cells, and endothelial cells as well, essentially creating these complex organs on a chip. Now, the cool thing happened when we understood that we don't have to mimic nature to 100%. We can essentially one-up it. We can create something better. In our case, it was bionic, what we called bionic microorgans. The idea is to take the tissues that we already create with tissue engineering and to add nanoparticles of that can sense oxygen uptake to the mixture, essentially embedding those particles in the tissue itself. So those particles are produced by a company called Colibri Photonics, and together we essentially managed to embed them into tissue in the bioreactor, allowing us to measure oxygen uptake rates in real time. And our tissues are very, very stable, and you can actually see that we can keep the tissues happy in the bioreactor for over 28 days, for over a month. Now, the interesting thing about these microparticles is that in the presence of oxygen, we see a phosphorescent decay in the fluorescent signal. Now, this means that if we expose the particles to a sinusoidally modulated amplitude light, we can actually get a signal that is, again, sinusoidally modulated back to us. Now, the difference in phase between what we sent and what we received is directly proportional to the concentration of oxygen in our sample. This essentially means that we are measuring phase shift rather than amplitude. Now, the reason it's important is because for the first time, this type of sensing paradigm is not sensitive to distance from the focus. For example, even when we are far away from the focal point, about 1.3 millimeters in the graph that you see here on the right, we can still measure the same phase shift. Why is this important? This is important because when we're doing high content screening, for example, we don't necessarily always get to the same point in the tissue. Sometimes we are a few microns away from the focal point. Now, if we were measuring amplitude, that would lead to differences in oxygen uptake rates, or at least measured oxygen uptake rate. But because we're measuring phase, we really don't care about it. The second thing that happens is that we can now measure for a long period of time. So even if half of the cells in our systems die and the particles drift away, It doesn't matter if we started the experiment with 10 particles, lost 5 of them, and then gained 2 more because of cell migration. We are not measuring amplitude, we're actually measuring phase change, and therefore we can measure it routinely without caring how many particles we have in the system. Now, what can we do with this type of measurement? Well, one of the things that we can do is start measuring kinetics of metabolic response to a drug, which nobody could before because all the other measurements are static. The first drug that we use is called rotenone. It's a commonly used pesticide, which directly damages the mitochondria. And indeed, the second that we add the drug, you can see that all the cells stop respiration at exactly the same time. So we do see a difference in the time to onset. So with increasing concentration of rotenone, there is a slight difference in the number of minutes it takes for the damage to occur, for at least for us to see it. But it is a very, very short time from the moment we add the drug to the moment that we see the response, suggesting that this is a direct effect of the drug on the mitochondria. And indeed, this is exactly what we see. The cells are dying from necrosis and apoptosis very reliably. A very different case is for an antiarrhythmic agent called amiodarone. Now, amiodarone is thought to cause lipid accumulation in the liver. And when you add it, you get this protracted response that seems to be dose-dependent. And indeed, when you look at the time to onset, the time it takes for the damage to occur from the moment you add the drug, you see that we are talking about anywhere from 2 to 16 hours until you start seeing damage from the moment you add the drug. This suggests lipid accumulation, and indeed, this is essentially what we're seeing. We're seeing that amiodarone causes both phospholipidosis and steatosis, essentially fatty liver disease in our system, like it does to patients in vivo. But the real proof in the pudding came with when we added acetaminophen. So acetaminophen, Tylenol, also known as paracetamol in some countries, is a very common pain reliever that is sold off the counter. And its toxicity, however, is the most common cause of acute liver failure in the Western world. And we thought that we knew why. For the last 30 years, we know that acetaminophen can be removed by glucuronidation or sulfation very, very rapidly. But when it's taken as an overdose, then different cytochrome P450 enzymes, CYP2E1 and CYP1A2 specifically, can break it down to a toxic compound called NAPQI, and that can damage the mitochondria. Luckily, we have glutathione in our livers, and glutathione can be used to conjugate NAPQI and essentially deactivate it. So only when people have fatty liver disease or alcoholic liver disease and glutathione is depleted, you're supposed to get damage because of NAPQI. Now here's the problem. The problem is that even though most people with overdose of acetaminophen appear in the ER and have liver failure, a small percentages, about 2%, actually have kidney failure. And then a smaller percentage has skin necrosis. And nobody could really explain why. You see, those two enzymes, CYP2E1 and CYP1A2, are not expressed in the kidney and they're not expressed in the skin. And NAPQI is way too active to diffuse from or flow from the liver to the kidney. So something else must be going on. What we found out is that from the moment that we added acetaminophen to our liver on a chip, you can see that we got a very, very distinct response. This is essentially all the cells in our liver stopping to breathe at the same time. There was no dose dependence. Essentially, it happened in a minute. The second that you add the drug, all the respiration that you see here essentially stopped. We had a second phase that was more linear. That was probably NAPQI production, but the first one couldn't possibly be a direct damage of NAPQI. And when you look closer into this, you can see that even at physiological concentrations of acetaminophen, like 1 millimolar, when you add the drug, you see loss of respiration. So we decided to dig deeper into this, and one of the things that we saw is that the effect is completely reversible, at least in the first 60 minutes. And then we went on to discover that acetaminophen was actually binding a protein on the electron transport chain, probably complex 3, and on its own, even without metabolism, it can block respiration in the liver. So this explains a lot of the side effects of acetaminophen that were not explained up till now, and could explain why people were getting kidney toxicity and skin necrosis. It does suggest some new warning labels for acetaminophen, and we published this work in Archives of Toxicology earlier this year. So using our system, you can get a very nice correlation to what happens in vivo. Across 4 different drugs, we get a beautiful response. The second stage of my talk, and I don't know how long I've been talking. John, will you give me more time?
Of course, of course.
So feel free to stop me at any time and ask questions.
No, very interesting. Keep going.
Yes,
okay.
Fascinating. Thank you.
So one of the interesting things that happens in drug toxicity is called an idiosyncratic response. Idiosyncratic response is essentially a response that happens not necessarily related to dose, but in 1 out of every 100,000, 200,000 individuals in the population will suddenly have toxic damage. And it's not really explained. It's one of the main reasons that drugs are withdrawn from the market after they've been introduced. Well, why does this happen? So usually, when we develop a drug, we're trying to find the doses for therapeutic benefits. So you can see this sigmoidal curve of concentration versus effect. And once we find it, then we can define a range of concentration, essentially the effective dose that we want to give to patients. After we define that therapeutic benefit, we measure the toxicity using all the models available to us. And hopefully that happens with much, much, much higher concentrations. Well, once you take this and you bring it to the market, then one of the things that happens is that patients are much more variables than laboratory animals or even cells on a petri dish. So you can predict that the toxicity profile in patients is going to be much worse for you. It's going to be much closer to the dose. So you need a significant margin of safety. And if you do everything correct, then essentially the patient will only get toxicity if he purposely or non-purposely takes a massive overdose, right? So you have a very nice safety. The problem with idiosyncratic toxicity happens when something is not known. You measure the toxicity and you see a certain response, but what's unknown to you is that the cells were actually placed under stress. And this stress profile happens at lower concentrations than you think. And then when you bring that drug to the market, then the patient population would essentially shift, and you can see that part of the normal dose suddenly becomes toxic. So this is a big, big problem. Essentially, we don't know why idiosyncratic toxicity happens. And one of the most common causes is thought to be immune-related because it's very difficult to mimic the immune response in rodents or in the tissue. So that could definitely be the case. However, another mechanism that is possible is sub-threshold effects of toxicity, essentially stress on cellular metabolic processes, for example, that might shift the effect down. So we decided to test that using our system. And we went to a drug called Troglitazone. And Troglitazone was developed as a TZD, as a drug for type 2 diabetes. And it was indeed released to the market. And only after it was released to the market, it was withdrawn due to idiosyncratic toxicity. And the mechanism of effect is still relatively unknown. So when we add troglitazone to our bioreactor, we see this pretty complex response. In some ways, it's similar to what we saw for amiodarone. So you see effect at the lower doses can happen in an hour or 2 hours. But on the other hand, it's kind of similar to rotenone as well. It can happen in as quickly as 30 minutes. And it's very difficult to imagine secondary damage happening in 30 minutes. That's very, very fast. So it suggests that there is something interesting here happening in the dynamics. And really, when you go into potential mechanisms, you can see that, well, it does cause cholestasis, essentially accumulation of bile acids in the liver cells that can ultimately cause damage. But on the other hand, it also increases mitochondrial membrane potential, suggesting it causes stress to cellular metabolic profiles, although this is quite mild. So what we decided to do is essentially try to track whether whether we can track mitochondrial function in real time. So to track mitochondrial function, we need to do two things. In addition to measuring oxygen and respiration, we also need to measure glycolysis. Essentially, the major route of taking glucose and bringing it into the cells and breaking it down. Well, when cells bring glucose, they can take it in through a process of fermentation that doesn't require oxygen, produce lactate, and this can produce ATP for the cells, essentially energy for the cells without oxygen. But this is very, very ineffective. Or they can take glucose and then burn it through the Krebs cycle, requiring oxygen but producing a lot more ATP. So one of the things that we thought is that if we have a drug that blocks the mitochondrial respiration, then we should see an increase in glycolysis. So we designed a more complex system by essentially connecting the entire thing to a microfluidic switchboard that allows us to take samples continuously and run it over glucose and lactate sensors. Now, these are commercially available, clinically relevant sensors that have glucose oxidase or lactooxidase embedded in a membrane. And in the presence of glucose or lactate, they produce hydrogen peroxide, essentially giving us a current. And we can measure this current. The biggest problem with this type of sensors is that they are not very reliable. And essentially, every measurement you make, you have to have a clear break. You can't continuously perfuse sample in a liquid medium because diffusion will cause the signal to spread out over the medium, and you'll measure something like a peak rather than a sharp step change in concentration. The second problem is that over time you get fouling of the electrode because of protein binding and because of and you're going to get salting of the membrane as well, which is going to cause a rapid loss of signal. So what we did is we devised a method to we developed this switchboard that allows us to address those electrochemical sensors very reliably. Essentially, what we're doing is we have a process by which we wash the sensors introduce an air bubble, and then the sample giving us a very sharp step change in concentration. And then we wash it again and run a calibration curve, a calibration measurement, to make sure that we don't have drift over time. And we can do it over and over and over for multiple days on multiple bioreactors using a switchboard. It looks roughly like this. So this is when we introduce the buffer, and then this is where we introduce the bubble of the air, and then this is when we introduce the sample. And you can see the measurements down here and how the actual system looks right below this. So this allowed us to really get some critical understanding on what happens when chemicals start damaging the mitochondria. So the first experiment we did is add rotenone again. That's the pesticide that directly damages the mitochondria. And what we thought would happen is that the cells would shift from respiration to glycolysis. And they do, but only for a few hours. After 3 hours of exposure to rotenone, glycolysis isn't sufficient. And essentially what happens is that the cells move to glutaminolysis, taking amino acids from muscle breakdown, for example, and start burning them up for energy. And this happens at concentrations of rotenone that don't really kill the cells, or at least not most of the cells. What happened when we introduced troglitazone was quite different. So troglitazone, we introduced it at 50 micromolar at a concentration that doesn't cause any type of damage at all. And you can see that the glucose uptake into the cell didn't change at all. But lactate production started going up, and oxygen uptake actually went down a little bit, suggesting that the cells actually shifted toward glycolysis very, very slowly. And essentially what we're seeing is that even at very low concentrations of drugs, concentrations that were predicted to be completely safe, we can actually see sub-threshold effect of compounds that are thought to cause idiosyncratic response, shown to cause idiosyncratic response. Essentially, they block mitochondrial respiration and causing the cells to shift toward glycolysis. And we can, of course, confirm all this using in static conditions. This allows us to really going back to our initial drawing. Essentially, this is the first time that we can use liver-on-a-chip technology to understand unknown effects, or essentially reveal unknown effects before we go to the market. And the only reason we can do that is because we can monitor function in real time, everything from oxygen uptake to glucose and lactate measurements. So this is it, and I'll stop over here. I'll recognize I have a very large group, and a lot of people helped me in the development of this technology, including Magnus Jäger from IBMT, and Sebastian Priel, who was his PhD students, and in my lab it was headed by a PhD student that recently graduated named Danny Bavli, as well as Gal Levy and Elishai Ezra from my lab. Thank you.
Very good. Thank you, Yaakov. A very good presentation, well illustrated, very relevant work, especially on a couple of fronts, the cost savings and the saving animal toxicity is really fantastic. And Gene, why don't you start things off with the questions? We both have them, I'm sure.
Yeah, I was wondering, what do you know about work out there to use real liver tissue to contain an organoid of some sort, and to use for these kinds of studies?
Well, the liver requires quite a lot of oxygen. So one of the things that you can do is use liver slices, and that's been done for the last 20, 30 years. You can keep liver slices for about 2 to 3 days outside of the body, as long as you give them a very high concentration of oxygen, about 90% oxygen. There is a professor named Eduardo Mitterrani developed slightly different technologies to take the liver slices and chop them into smaller pieces, 200 micron cubes that they can keep going for longer.
Mm-hmm.
And then there are other technologies using tissue engineering, like Linda Griffith's liver chip, and of course Organovo is now 3D printing liver tissue in not very good resolutions. It's a more complex architecture. The problem with all these technologies is not that they don't mimic the liver physiology or function. They do it not bad. They do it well, but they're not measuring fast enough. So they're not measuring effects in real time. They are replacing an animal experiment with an in vitro experiment, but they're doing the same type of endpoint assay.
Mm.
So if mice, like you stop after 24 hours and measure what happened, that's very nice, but what happened after 1 hour is much more relevant to what happened after 24 hours.
Mm-hmm.
And our system is the first to be able to measure things in real time. We can measure things in minutes and over long periods of time. And that kinetics tells us something about mechanism of action and adverse outcomes.
Mm-hmm. Early on in your talk, you were discussing how the liver has kind of, it's not uniform. It has parts throughout, and it functions somewhat differently. Now, a small device like yours, can it replicate, a large organ like that has more complexity than, that has a large scale complexity?
Right. Well, the complexity of the liver is actually starts and ends at about 250 microns. That's the length of a single sinusoid. It's a very large organ, right? It's pretty big in humans, but that organ can be subdivided, and essentially all the blood that goes into the liver goes from very large blood vessels into smaller and smaller and smaller blood vessels. There's no function there.
Mm-hmm.
Only when it enters a capillary, a sinusoid, then metabolic function starts. Now, metabolic function starts at the beginning of the capillary and ends at the exit of the capillary. And that entire distance is 200 microns.
I see. Okay.
So all we need to do is mimic that distance, that 200 microns.
So it is consistent throughout the entire organ. It's
Yes.
made up. Okay, I see. Sorry, I got the wrong impression there.
Okay, and let me just comment to the audience. And anybody has a question, you're free to ask. I just have a couple of questions, Colby. I guess these studies probably are well funded, because I would imagine animal groups like PETA and the pharma industry would be lining up to fund them. Is that true or no?
Well, I certainly didn't receive any money for PETA. No,
well, I would think they'd be very interested in this, because it would spare animal, experimenting on animals, obviously.
Do they sponsor any research? I wonder. I think they're an advocacy group. I don't know if they sponsor research.
I don't know if they sponsor any
Okay.
research, but quite frankly, I have to tell you that I'm not against animal research. I think animal research is a critical engine of medical and biological discovery.
Okay.
I do want, however, to eliminate animal use in industry, especially in toxicity studies, because this is where it's not really relevant, and we can very rapidly find alternative tools. Now, we're talking about the majority of animals used actually in industry, so that's going to be a massive chunk, but I'm not for eliminating animal experiments at all. I mean,
Right.
I have a sister with type 1 diabetes, and without animal experiments, she would be dead.
Right.
Right?
Well, you mentioned
that insulin was discovered on experiments on dogs.
Well, you mentioned the cross-toxicity, different toxicities for different species. And thalidomide, I didn't realize that was a shortcoming of those trials before. Thalidomide was because they were toxic. They were toxic in humans, obviously, but not toxic in mice.
Nope, they're not toxic in mice and rats. Very safe. The other thing that is very, very interesting is that aspirin, very safe in humans, completely toxic in rodents.
Okay, that's interesting.
Yeah.
Well,
So
there
is a big lack of correlation. There are a couple of reasons for that. One of them is that there isn't one-to-one homology in the different enzymes, the cytochrome P450 enzymes that break down molecules in human and in rats. CYP3A4, for example, which is an enzyme that breaks about 30–40% of the drugs on the market, is not expressed in rodents. The rodents have a CYP3A1, which is a variant, but it's not the same enzyme, and it cuts the molecules slightly differently.
Mm-hmm.
So that's one of the reasons for the differences. There are many more.
Do you have any comments on liver cells being produced by 3D printing versus stem cell?
Liver cells are not produced by 3D printing. I think tissues are built by 3D printing.
Okay.
So we just published a paper in Nature Biotechnology showing the first massive expansion of primary human hepatocytes. This was a very big breakthrough in the field. We can now have highly functional human hepatocytes. The other thing that we published in July is the first derivation of functional hepatocytes from embryonic stem cells and genetically induced pluripotent cells. And this is something that the field has been struggling with for many, many years. We've been able to differentiate hepatocytes for the last decade, but those hepatocytes, liver cells, have had little to no metabolic function. They were really behaving like fetal hepatocytes before the baby was born. So one of the interesting insights we had in July is that, actually it's an insight after my last child was born, that the microbiome might actually affect liver development. And we essentially found out that there are two molecules produce a variant of vitamin K called MK4, and the other one is lipocholic acid, a secondary bile acid, that the liver will only be exposed to them after birth, that play a critical role in waking up all the cytochrome P450 enzymes in the liver. So this was just published this July on the cover of Hepatology, it's the first time that we actually were able to do a one-to-one comparison between primary human hepatocytes and embryonic stem cell-derived hepatocytes and show a similar toxicology profile. We got a 97% accuracy in the prediction of toxicity. So I think the field as a whole is getting much, much closer to the point where we can produce hepatocytes from stem cells. And we just surpassed a major hurdle in the ability to expand human hepatocytes. These are two competing technologies. And I'm pretty certain that both of them will be used in some various level of function by the industry in the coming years.
You know, stem cell science is rife with controversy. Are there any ethical issues at all involved, or no? With using
in Israel,
or is that a stupid question?
No, it's not a stupid question. I mean, human embryonic stem cells are produced from discarded embryos that are prepared during an IVF procedure. In the U.S., there was a bit of controversy during the George W. Bush years because of the idea that they are destroying embryos by producing those cells. This is true, but if you wouldn't produce these cells, we're talking about embryos that will go to the incinerator anyway.
Okay.
So most of the world, England, Israel, that wasn't an issue. I think the governments and even religious officials realize that IVF is becoming very, very common. In the procedure of IVF, we produce a lot of fertilized embryos, and we implant only so many until a successful pregnancy, and then we discard the rest. So instead of discarding, we can generate stem cells out of them. The deal is by the time that this argument was made its way to Congress, it was no longer needed because the Yamanaka protocol emerged, and we can now genetically induce stem cells from skin cells very reliably, very easily.
Mm-hmm.
So we no longer need embryos, really.
Hey, has this new technology ever been incorporated in a real trial yet, or is it strictly experimental?
So I can tell you the technology was developed in tandem together with L'Oreal and Cosmetics Europe, and right now we're in the process of a trial run at the Hebrew University of several compounds. Hopefully, we'll take it further from there. We just quite frankly, look, we just published it in July, the Archives of Toxicology paper on the toxicity of acetaminophen, and we have another paper coming very soon on the metabolic profiling of Troglitazone, and so this is very, very recent. You got it right out of the oven.
Very good. Well, Yaakov, I'd like to thank you very much for spending the time. And Gene, do you have any comments before we sign off?
So far, this was great. Thank you, Yaakov. Fascinating discussion. Thanks for coming to MedGadget.
Very good, Yaakov. We welcome you to come back anytime for more presentations.
Thank you, Dr. Bennett. It's my pleasure.
Okay, we're going to end the broadcast. Hold on, Yaakov.